Alright, here we go. So this is going to be aerobic respiration. Let me know if I go off the paper, it will happen. And that way I can get back on. So aerobic respiration, this is the process of getting from glucose with oxygen. And we're going to be generating carbon dioxide and water and a release of free energy. And that release of free energy is going to be used to make ATP. So that's the overall process that we're going to be looking at. Okay, here we go. 4 major steps. What are they? This one. Glycolysis, then citric acid cycle, then, and finally, in that order. So that's the order we're going to be taking. Okay, so we start off with some glucose or some other large biological molecule. So I'm just going to put in parentheses up here food. So something that you've consumed that has stored chemical energy in it. Okay. Now, this is going to be my blue section, so I'm just going to outline this whole thing in blue. And we will be writing our steps of glycolysis here in blue as well. Glycolysis is a really long process. It has like 10 steps, and there's a lot of stuff going on and a bunch of intermediates, and then there was stuff about an energy investment and energy payoff in your book, and you're probably like, well, what am I actually supposed to know about all of this stuff? And that's what I'm going to tell you. There are 3 major things that I want you to know about glycolysis. Okay. First of all, overall, what glycolysis is the oxidation of glucose into how many pyruvates? 2 pyruvate molecules. What does it mean to oxidize something? No. Lose electrons. You are removing electrons from it. Electrons are going to be the main source of energy in this process. So anytime that I say that we have oxidized something, that means that we've taken away electrons from it. And then if you oxidize something, that allows you to then reduce something else. What does it mean to reduce something? To add electrons to it. So we're taking electrons from this thing and adding electrons to this thing. Those things that are carrying electrons are going to be the NADHs and the FADH2s. They transport and carry high energy electrons for us. Okay? So this entire process, the glycolysis and the citric acid cycle, is all going to be about stripping electrons out of glucose and then transporting it to the electron transport chain. All the stuff that we're about to go through is dedicated to that. Yes? Blue is glycolysis.
So they just all stand for all different stages.
I'm going to break up each stage into a different color, and that way you can know, you'll be able to trace back like where certain things are coming from and what occurred when. Okay, so step 1 that I want you to know about glycolysis is occurring right here, where we generate some ATP. Okay, so one thing that I want you to know about glycolysis is that we have a net gain of 2 ATP. So net gain of 2 ATP. Now we have that because in glycolysis we have to invest 2 ATP up front to start to break down, to start to fully oxidize the glucose into pyruvate. But then on the back end we generate 4 ATP. So 2 go in, but 4 come out. So we have a net gain of 2. Right? Okay. Yeah, it is a good deal. But it's not very much. It's not enough, but hey, it's something, right? So up here, anytime that we generate ATP, because there's 3 main places that we generate ATP in this process. And I want you to know when those 3 main places are. This is one of them. We're going to get ATP. Now, there are 2 different types of phosphorylation. What are they? Ooh. There's oxidative phosphorylation, which we'll be doing at the end, but this is not oxidative phosphorylation. This is the other kind. What is it? No. No. Substrate level. Substrate level phosphorylation. And how many ATPs do we get here? 2. Net. Because 2 went in, 4 came out. So we ended up with 2. Okay. So this is going to progress us down glycolysis. So we're moving down, we're moving down, we're moving down. And then we reach where something else is going to happen. So then we get to step 2 right here. So net gain of 2 ATP is happening here. So now step 2, which is here on the diagram, we are going to write here what is happening. Okay. We are going to produce 2 NADHs. And these are going to enter, from now on, I will never say the words electron transport chain. Because why say many word when few word work better? Is that what it is? Yes, exactly. So I'm going to be instead be saying ETC for electron transport chain. Okay. So we are producing 2 NADHs, which are going to enter the electron transport chain. This is done through the oxidation of this glucose. We are removing electrons from the glucose and we are transferring those electrons to an NAD+, which reduces into an NADH. So basically all that means is we just gave NADH, we just gave NAD+ electrons, and that changed its name because of chemistry, and we don't care about that because we do chemistry. And then that NADH is going to move to the electron transport chain. So this is a long arrow. You ready? Here we go. Arrow, 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 all the way down. Here it goes into the mitochondria. All the way, it wraps around this stuff. There's a hole in there somewhere. All the way down to the electron transport chain. Yeah. So this is the first place that we are delivering electrons to the electron transport chain. Again, the entire process of glycolysis and the citric acid cycle is solely to get these electrons to the electron transport chain by driving them in these NADH and FADH cars.
Yeah.
ETC.
Kind of like
Okay, so that was step 2. Then we end up with step 3, which is the third and final thing that I need you to know about glycolysis that occurs here. And here we have broken down glucose into pyruvate. So step 3, 2 pyruvates. Are produced. This whole process occurs outside of the mitochondria. All right, at this point, one of 2 things can end up happening. This is where the split occurs. So we have our pyruvate. What's the next thing that must be determined? Is there oxygen? Is there oxygen? All right. If there is not oxygen, what would this pyruvate be used in? oxygen, there's not oxygen, what happens?
Anaerobic.
Fermentation.
Maybe some fermentation.
It is. Fermentation, anaerobic respiration occurs, and that's what your assignment tonight is over. We don't care about that, because what if there is oxygen? If there is oxygen, then what we're going to see next is the active transport of pyruvate into the mitochondria. And that is going to be step 1 of the Krebs cycle. Now, your book doesn't do a great job of delineating between the end of glycolysis and the beginning of the Krebs cycle. There's actually a reaction called the link reaction, which stitches these two processes together. And what we're about to look at is technically part of the link reaction, but we're going to consider it part of the citric acid cycle because your book does. Okay. Yes, so now we've moved to red. Okay. So next step, we're going to see pyruvate enter the mitochondria. Ew, no, it's gross. Okay. The citric acid cycle banner is way over here. You ready? Here we go. It was a long ride. All right. Pyruvate enters the mitochondria via active transport. Does that require an input of energy? Heck yes, it does. So here we have to use a little bit of that ATPF we've already gained. Ooh. But it's okay, it's going to pay off in the end. Just you wait and see. So that's the first thing we need to see is we have to get the pyruvate into the mitochondria. That requires energy because pyruvate is really big. It doesn't go through the membrane very easily. We got to pump that stuff across. Okay, so now we are here. We have pyruvate into the mitochondria. Now, one thing that I'm sure you remember from your reading over Chapter 9 that you've already done and spaced out evenly so that you didn't get stressed tonight is that pyruvate, once it enters the mitochondria, is not yet ready for the Krebs cycle. 3 things must be done to it. I would be so impressed here. What 3 things need to happen?
Wait, say the question.
What 3 things must be done to pyruvate before it can actually enter the citric acid cycle? This is technically called the link reaction because it is preparing the pyruvate for the next step. But we're adding the CO2 does leave. You probably saw that here, right? That's awesome. That's good. So we're going to move down here. Okay. This is technically our step 2 right now. And what we're going to see is carbon dioxide is going to be released. Now that is just so odd. It's almost like whenever you guys learned the equation for cellular respiration, you guys talked about carbon dioxide being released. This is the first place. This is the first place that CO2 gets released right here. Okay? So let's go ahead and write down what's happening. Number 2 here, it is the removal of a carboxyl group. As CO2. So the carboxyl group leaves as a molecule of carbon dioxide gas. And we have to exhale that later. Right? Of carboxyl group as CO2. I'm speaking without any kind of whatever those words are. 2 goes here where we spiral off our CO2. Okay? Next. Step 3 is happening right here. We are going to generate some NADH. Yes, that's good. That's like the goal of this whole process. We're trying to crank out NADH. NADH is reduced whenever the pyruvate is oxidized. Oxidation reduction. You must oxidize something in order to reduce it. Okay. So then step 3 down here, what we end up seeing is oxidation of pyruvate. Is going to generates NADH, which then moves to the ETC, the electron transport chain. All right. So that's good. Now we have more NADH headed in. We're dropping off electrons. We're stripping electrons out of pyruvate and dropping them off. And that's the overall goal. We're trying to harvest as many electrons out of this original glucose molecule as we possibly can. We're just stripping them out. Okay? We're taking them out and dropping them off. All right. So now, the citric acid cycle can almost be started, except the problem here is that once we've broken down pyruvate to this level, we have something called acetate, which cannot quite be used in the citric acid cycle because all of the enzymes that start the citric acid cycle require a coenzyme to get it started. And that coenzyme is called coenzyme A. Right? So in order for it to bind properly to the first enzyme of the citric acid cycle, we have to have coenzyme A attached to our acetate. And when coenzyme A is attached to acetate, it receives the name acetyl coenzyme A. And that's where we are at next. So, step 4. Coenzyme A is added, which then equals acetyl coenzyme A. A bunch of words. Basically, it's just an intermediate that's required to make in order to start the Krebs cycle. This whole thing, 2 through 4, is actually called the link reaction. There is more stuff we're going to add. It's technically not going to be a 5, though. Not in the box. I don't expect you to have any room left in the box. We're going to go below the box in the margin. Really? That's impressive. Link reaction, RXN reaction. Oh, well, that's a problem. Yeah. Reaction, RXN.
Where's the reaction?
Oh. All right. So now let's move on up right here. All right. So this acetyl coenzyme A that is now produced is going to enter the citric acid and or Krebs cycle and or TCA cycle. It has a bunch of names. And I'm just going to shade this whole thing in. As we go through this cycle, there are a litany of steps, and it's really confusing and flow charts like crazy, and it's a whole bunch of stuff that we don't need. But what I do need you to know is that as you move through the citric acid cycle, it is a series of oxidative steps. So we are stripping electrons out of this acetyl coenzyme A, which started off as pyruvate. And every time that a reaction is done on it, it actually becomes something else. And that's why there's, a million words within the citric acid cycles, because every time something's done to it, it gets renamed. And it's really confusing. You don't need to know all that. But what you do need to know is that this whole time we are oxidizing the molecule that's moving through the citric acid cycle, which started off as acetyl coenzyme A. And every time we oxidize it, we are going to be releasing carbon dioxide. Just like we saw carbon dioxide being released at the beginning. So carbon dioxide is coming off. If we oxidize something, then that means we're able to reduce something else. What do we reduce? We reduce the NAD pluses into NADHs, which then go to the electron transport chain, the ETC. We reduce the FAD The FAD pluses, FADH plus. Anyway, we reduce those down into FADH2, and those transport electrons to the ETC, the electron transport chain. So again, the entire process of glycolysis and the citric acid cycle is we're taking this glucose, we're stripping as many electrons out of it as we possibly can, and then we're transporting them to the electron transport chain. We're just storing up a bunch of electrons. The big payoff has not come yet. We're getting it ready. We're building up a big pile of electrons. Okay? Now, to kind of wrap up the Krebs cycle, there's 2 points that I want to put over here. Okay? You have the margin. Come down here below. Really? Oh no. All right, you might have to go to the back or something. Yeah, you might have to go to the back. But you can't go to the left because we're going to start using all this. See, this whole thing's going to get used up down here. You can do the stuff at the end on the back. Though. That's okay. You can do the stuff on the end on the back. Once acetyl coenzyme A is formed, the Krebs cycle focuses on generating NADH and FADH. Okay, that's thing 1. Thing 2. This is all going to run together if I don't separate it. The Krebs cycle harvests. E minus stands for electrons. So harvests electrons from pyruvate and transports them to the ETC. That is the primary function of the Krebs cycle. We're harvesting electrons and moving them to the electron transport chain, which is where we are headed next. All right, we have now at this point generated this big mountain of electrons, and it's about to pay off because now it's time to make some ATP. Here we go. Okay, electron transport chain, the ETC. I've now moved into purple. We're gonna have to pick up the pace a little bit here because we're running out of time. Yep. Last part, it says the Krebs cycle harvests. Who am I talking to right now? Sasha. The Krebs cycle harvests electrons from pyruvate and transports them to the ETC. Well, I got to keep going, but I'll get it back to you here in a second. Okay. Yep. We have to get through it though if I'm going to upload it. Okay, so we now have all of these electrons that have come into the electron transport chain, and this arrow is now going to become purple. Wow. I know, it's amazing. Like, even in your side of your cells, everything just turns purple. It's incredible. I know, it's wild. It's red
and blue turn purple.
That's exactly why. It's crazy. Okay, so the electron transport chain, your book has a beautiful image of this. So what's going to happen is the electrons come in to the electron transport chain, which is a series of membranes, transmembrane proteins that are embedded in the inner membrane of the mitochondria. And what's going to happen is the electrons are going to be forced to move down this series of steps all the way down these membrane proteins, and while they're moving, they're releasing just a little bit of energy at a time. And that little bit of energy can be used to do something. And what they're going to do is push hydrogen ions to the other side of the inner membrane of the mitochondria. And this is going to set up a concentration gradient of hydrogen ions that we can use to do something. That process is called chemiosmosis. But this is the figure from your book, and it's really good. So this is what it shows. It shows the NADH and the FADH2 delivering electrons into the electron transport chain. In come the electrons. Okay? Those electrons are going to be pulled down this series of enzymes, which causes it to fall through a series of reactions, and it moves from higher energy to lower energy. And they're actually set up like this, to where as you move down, you're moving down to lower energy level, and they're actually being pulled. They're being pulled by a molecule that has a really high electronegativity. What molecule is that? That's pulling these electrons through the electron transport chain. Oxygen. Boom. Oxygen here. So why do we need oxygen for aerobic respiration? This is why. This is where it comes in. That's why it's glucose plus oxygen. Oxygen is the final electron acceptor. It's pulling the electrons that have been dropped off from the Krebs cycle and from glycolysis through this series of reactions, and then sucking up all of the electrons at the end and generating. Boom, that's where the water comes from. I know, it like all makes sense now. So let's go back here. Okay.
It's like a president getting there in a hurry.
So we have electrons, they tumble through that series of membrane proteins, and we'll talk about what they're doing here in a second as they tumble through. But they're being pulled towards this oxygen molecule here. And then as the oxygens receive these electrons, they're going to start bringing in hydrogens as well, because positive and negative charge. Neat, right? And that is going to generate water. Here's our water. No, it's darker blue for me. You guys can do whatever you want with that right there. All right, so we end up making water, and that's where the water comes from. Okay? All right, so now what we have done is we've sent electrons through the electron transport chain, and I need to tell you now what was going on during that time. So, number one, right here. Number one, right here. Okay, try to stay with me. Electrons from, I know this is a lot, NADH plus FADH2 are pulled through ETC by oxygen. Not here, no. The reason why oxygen is able to do this is because it has a high electronegativity. And that causes it to be sucking up electrons, so it's physically pulling it through the electron transport chain, maintaining a one-way flow of electrons through the electron transport chain. Okay. So then number two, what is this actually doing? Well, this is a process which is, as the electrons move through these membrane proteins, they are going to be doing actual work, and they're going to be pumping hydrogen ions to the other side. So these are just hydrogen ion pumps. It's going to be pumping hydrogen ions to the other side of this inner membrane of the mitochondria. So number two is happening right here. And I'm going to be saying here, H plus is just a hydrogen ion. So H plus is pumped across the mitochondrial membrane. Mitochondrial membrane. And this sets up a concentration gradient of hydrogen ions, and this is where the magic happens. So we've used all of these electrons that we've harvested from glucose, and we've pumped a whole bunch of hydrogen ions to one side of the membrane. So now we have a really high concentration of hydrogen here. And then, look up here, this is important. So this is that inner membrane of the mitochondria. There's an enzyme here called ATP synthase, which you read about, okay? ATP synthase is going to be creating ATP out of ADP and phosphorus, okay? So this is what's going to happen. These hydrogen ions are going to fall through, it's like facilitated diffusion, they're going to fall through ATP synthase to a lower concentration. And as they fall through, they drive the ATP synthase machine and generate ATP. It's a really cool process. No, I don't have time for that. But, yes, US. Yes. Yes. Okay, sorry. So where was I? Yes. So now we need to move on to step 3. Step 3 is happening right here. We're still on purple. This moves on to oxidative phosphorylation. Because oxidative phosphorylation is a part of this process. What?
4 in the orange. Should I use it?
4 in orange. I wasn't. Orange didn't have a 4. Orange is just what I use for ATP.
No.
I used red.
Orange. Oh, that is a red.
That's where we add acetyl coenzyme A. Okay, number 4. I'm sorry, not 4, 3. Nick kept saying 4.
Sorry.
3. Number 3. The H plus gradient is used to power ATP synthase, which is an enzyme that takes ADP plus P and generates ATP. Okay. This is the big payoff. So then on number 4, which is now going to occur right here, ATP is generated through oxidative phosphorylation. And this is what we end up with. We're going to get 32 to 34 ATPs here, depending on the internal conditions within the cell or within the mitochondria. For the entire process, this would correspond to 36 to 38, depending on the conditions. For the entire process. So that's all occurring right here. We got this ATP. Yay, energy. That's the big payoff. Whole bunch more than glycolysis. Glycolysis, we got a measly 2. Krebs cycle, we get 2. Boom, right here with oxygen, we can generate 36 to, or 34 to 32, depending on the conditions. Okay, so that is technically the end of the general process. Now, what I want to try to do, and we don't have much time to do it, is just kind of wrap this whole process up. So just bare bones, what I want you to know about these processes. If we look at glycolysis, I'm going to tell you this while I'm writing because the bell's going to ring on us. Last hour we got through this whole thing, so I'll post their video and it'll have the whole thing on it. Okay. So move down here to the bottom for me and we'll go as far as we can here. In glycolysis, the main thing I want you to know is we are going from glucose to pyruvate. After glycolysis, we are going to enter the Krebs cycle, or the citric acid cycle. The main thing I want you to know from that is we are generating NADH and FADH2 for the ETC. So many abbreviations. for the electron transport chain. That is the major process in the citric acid cycle. After the citric acid cycle, then we move into the electron transport chain, the ETC. Oh no, I should have done that in purple. Oh well. There we are going to set up a hydrogen ion concentration. And then we move into oxidative phosphorylation. Oxyphos. That kind of sounds like a real thing. And in oxyphos, so in oxidative phosphorylation, we are going to use the hydrogen ion concentration to drive ATP synthase, where we make 32 ATP. 32, 34, 33, somewhere in there, ATP. So that's the general overall process. What time are we out of here? 39, so I have 1 more minute left. I'm going to move up here real fast. Let's do this real quick. Look at the original equation. Where is the glucose used? Which of the 4 processes? Where do we find the glucose? Glycolysis, so I'm going to underline it in blue. Where does the oxygen come in? Remember, it's at the end of the electron transport chain. Okay. Where is the carbon dioxide released? Citric acid cycle. Where is the water released? The electron transport chain as well. Okay. I will be posting 6th hour's video